Dear students, I am Dr. Nidhi Sharma, Associate Professor Zoology, p.g.govt.college, Jhalawar, Rajasthan. Today, I shall tell you something about genetic recombination in short. What is genetic recombination? Genetic recombination is exchange of genes between two DNA molecules. This forms new combination of genes on a chromosome along with mutations. Genetic recombination contributes to genetic diversity. Now, the types of genetic transfer. First is the vertical gene transfer. This occurs during reproduction between generations of cells. Second is the horizontal gene transfer. This is transfer of genes between cells of the same generation. Now, what are the mechanisms of horizontal gene transfer? Genes are naturally transferred between bacteria using three mechanisms: transformation, second is transduction, and third is conjugation. All three result in recombination of genes. Now we will talk about transformation. Transfer of naked DNA from one bacterium to another. It works best when donor and recipient are closely related. Transformation was discovered by Frederick Griffith in 1928 while working with Streptococcus pneumoniae. Griffith's experiment is shown in this figure. Here you can see two bacterial cells. Now, in the first stage, living encapsulated bacteria were injected into the mouse, and living non encapsulated bacteria were injected into the mouse. In the second stage, the mouse died, which had been injected with encapsulated bacteria which were living. The second mouse, which had been injected with non encapsulated bacteria, remained healthy. Then, in this third stage, colonies of encapsulated bacteria were isolated from dead mouse, and a few colonies of non encapsulated bacteria were isolated from mouse. Phagocytes destroyed the non encapsulated bacteria. Then, heat killed encapsulated bacteria were injected into the mouse, and living non encapsulated and heat killed encapsulated bacteria were injected into another mouse. In the first case, where heat killed encapsulated bacteria were injected, the mouse remained healthy. No colonies were isolated from this mouse. Where living non encapsulated and heat killed encapsulated bacteria were injected into the mouse, the mouse died, and the colonies of encapsulated bacteria were isolated from the dead mouse. Now, there was another experiment by Avery, MacLeod, and McCarthy, which showed that DNA was transforming agent. Competent cells pick up DNA and integrate it into the chromosome. Here in this diagram, we can see a the last stage, the genetically transformed cell. It starts with DNA fragments from donor cells. You can say capital A, B, C, D, E, B, C, F, etc. Here, the recipient cell has a chromosomal DNA with the genes small a, b, c, d, and e. Now, the DNA fragments are injected into the with the recipient cell, and now in the first stage we have recipient cell which takes up donor DNA, shown by the figure where you can see the donor DNA with capital AB genes and the circular DNA, which is the recipient cell's circular DNA with small ABCD genes. Now, in the second stage, recombination occurs between donor DNA and recipient DNA. Here in this, you can see the degraded unrecombined DNA, and recombination occurs between the donor DNA and recipient DNA. In the circular DNA, you can see the A, B, C, and T DNAs of the recipient, and the capital B being the DNA of the donor, which is got incorporated into the bacterial DNA, and the cell becomes genetically transformed. Now we come to the process of transduction. In transduction, bacterial DNA is transferred from donor to recipient via bacteriophage. A bacteriophage is actually a virus which infects the bacteria. Two types of transduction are found, which are generalized transduction and specialized transduction. Now, this is a diagram showing transduction. Here in this diagram, you can see the recombination. In the first stage, a phage infects the donor bacterial cell. The second stage, the phage DNA and proteins are made and the bacterial chromosome is broken into pieces. In the third stage, occasionally during phage assembly, pieces of bacterial DNA are packaged in a phage capsid. Then the donor cell lyses and releases phage particles containing bacterial DNA. Fourth stage, a phage carrying bacterial DNA infects a new host cell, the recipient cell. And in the fifth stage, recombination can occur, producing a recombinant cell with a genotype different from both the donor and the recipient cell.